Good morning or good afternoon, everyone. And we are going to talk about local ancestry prediction. My name is Tatiana Tatarinova, and I have three helpful ladies here, Dallas, Amanda, and Jolene. So they are a part of summer bioinformatics program, and they will help us learn about local ancestry. This work was done in collaboration with Nikita and Alexandra, who are my students and collaborators. And so we have developed this algorithm. It hasn't been published yet, but we have already used it for several projects analyzing data from around the world. Okay, so of course, we are all mutts. And if you sequence DNA from any person, it will be very hard to find somebody who is a pure something. So because people move around, people are not docile, people are not bound, not chained to location. People marry people that different from different villages, from different cities, from different countries, and this is healthy, this is good, but it makes genetic identification challenging because any genome, for example, here, if you have a look genomes of from Latin America, we find parts that are European, parts that are Native American, parts Asian, and for example, somewhere in Peru, we have lots of admixture from Japanese communities, and it's good, is healthy. Why it is difficult? What kind of problem it presents? For example, if you want to study association of any position of the genome with certain ancestry, certain population history, we would like to take, for example, pure African. And we'll look for pure African, say, in the United States. We won't find pure African in the United States because people are mixed. If you want to find a pure Russian, for example, is even more cryptic. There is no such thing as pure Russian. It's all hodgepodge and mixing of Slavs, Genghis Khan descendants, and you name it. And since people were moving around a lot and exchanging DNA either in peaceful way or by rape or just any way, it is very hard to trace the origin of a particular spot and attribute population process and associate with a gain function or loss of function adaptation, for example, to certain altitudes. So that's why what we want to do, we want to because finding, of course, one approach would be to find people in the remote villages that, for example, a mountain in, say, in Tibet, people who live there for centuries, they haven't exchanged their DNA with anyone. And then, yes, that would be one possibility, but how many locations that were untouched by civilization, untouched by human migration there are? I would say very few. So the only reasonable way is to not being very picky when you collect the data. Of course, use some metadata, but try to find either a way, looking at the genomes of admixed individuals, find areas of pure ancestry, and then use them for all downstream studies to find association between population history and certain adaptations to altitude adaptation, temperature adaptation, and you name it. And of course, if you think about it, it's not only problem for humans, it's problem for any studies for any large population. So that's our motivation. And of course, we know people are of course mixed, and so people are all related to each other, and as you know, representation of people as a tree is a bit misleading. And ladies, now I'm going to pick on you, and sorry to put you on the spot. Why do you think representation of people as a tree is misleading?
Yeah. I say, like, for one thing, it is trees can be independent of one another.
Yes, and Mandora is very helpful here. Yep. So,
mute yourself. Just put yourself on mute. Now,
so, Jolene, why do you think? Oh, I have to say it again.
Yeah.
Yeah, the dogs are in the way.
So, for one thing, with the trees, it assumes that it's just a clear linear path, so that if you're looking at one region or one area, then it's going to stay that one region or one area for each generation.
You're asking me why, or?
No. I was saying, like, if you had anything to add besides that.
I mean, it's kind of confusing for me, just because when I think about the lineage, if we're thinking about how all those different relationships could happen where, as Tatiana was saying, you get together with someone of an opposite race or rape, and she said rape, but anything like that, I feel like that lineage would not still be straight. And if it is straight, then it means you're not being accurate in data recording,
Right.
or anything like that. So, I mean, that's the part that confuses me. It could still be straight if you were just looking at those of that same lineage, but I just
Right.
don't think that's accurate in genetic data sequencing.
Yeah. So tree is a simplification, and one of the examples where trees are not being very useful. For example, if you look at the tree of when our ancestors and Neanderthals separated, so they were in as a tree, like two branches. But of course, as you know, there were integration from Neanderthals to modern Europeans, and of course, and it stopped being a tree because there is also a horizontal branch connecting it, so it's not really a tree. And if you look at people say from Africa and they people from Asia, and they don't actually live in separate branches because there are lots of links, lots of marriages, lots of DNA exchange between separate branches of the tree. So of course, tree is a convenient simplification, but it's really extreme simplification. So what do we do in order to describe our genetic profiles? So we use ancestry informative markers. And ladies, do you remember what are ancestry informative markers?
Anything that marks where your ancestors came from and how it all got started, basically.
Yeah. So in essence, you are right, but the best ancestry informative markers on the genome, it's a SNP. So a SNP has different frequencies in the population A and population B, right? For example, in extreme case, so all individuals in one population have allele, say A. In population B, everyone has G. That's going to be ideal informative marker to separate A and B. But since there are only 4 nucleotides, and we want to separate many different pairs of populations. So in this case, it becomes an optimization problem, how you choose a set of informative markers in order to be able to identify all possible pairs of populations. And sometimes it's very easy. If populations are very far apart, it's very easy to find markers to separate them. But if populations are very close together, it will be very hard, and sometimes it will be plain impossible to find. So and those of you who took my computational biology class remember that we used admixture in order to represent individuals, and so that's of course usually the first step of all the population studies. You collect SNPs either from DNA chips or either from whole genome data, and then you do admixture, and you look at admixture profiles and how admixture is supposed to look. So first of all, if you look at say one population, look at say Greeks here, they are supposed to be looking very similar, but they're not supposed to be identical, because if they're identical, it means somebody cooked the data, somebody would maybe sequence one person 25 times or 100 times, just being lazy, or took data from relatives. That's also you're not supposed to do. You're supposed to do sample around population. And also, if you look at Greeks, you look at other Mediterranean populations, for example, Italians, Bulgarians, things supposed to look change from one population to another continuously, because political boundaries are just political boundaries, and if maybe during the 20th century, they were quite restrictive. Maybe in 19th century, they were not that much, and people were marrying, across the border quite easily. So you would expect, rapid change in composition on both sides of the border. So borders are transparent, and now in the European Union, people will be mirroring each other very freely. So, and that's what you see, and that's your first QC that's you're supposed to do when you look at all the data. So, and then so you identify your populations, and from when you're happy with the way data looks. So what you do, you create what is called zombie populations, and zombie populations are putative ancestral populations, and then they serve as you decide what dimensionality you want for your problem, and the zombie populations serve as a reference, as a basis, and then you project all your newly calculated data on this basis. And and then the next step of the approach, you take some data, you project all the individuals, you collect your average, calculate admixture profiles, and then now you have a representation of a typical, for example, Chinese. You have representation typical Russian or typical Tatar, and then you can, if for example, if a new person comes along, then that's one that's denoted here as X, and you assume that everyone is continuously, genotype changing continuously, and of course, in this case, you ignore rivers, you ignore oceans, you ignore everything. You assume like a continuous approximation of genotypes. Then you can place a person on a globe based on the values of those components. So in this case, it's a way to reconstruct the position from the person. Or if you have a person who is, say, you're doing some forensics DNA analysis, you have an unknown body, you can look at the DNA, and the first thing, the easiest thing to do is to do this kind of admixture analysis, and you can, it will tell you where the person is likely from. So, but of course, it's going to be where his ancestors are likely from. The person himself or herself may be born totally in different parts of the world. For example, like I am from Russia, and of course, I'm also a Mat, and my daughter was born in the States. And of course, like your DNA would be more similar to DNA of similarly admixed people in Russia, but it will not reveal her actual birthplace. It will not reveal her actual latitude and longitude where she is located right now.
So in this chart,
yeah.
Is it all relative location, or is it another value?
So in this case, you usually take here or which one? This chart, which one?
The one at the bottom.
Okay, so this one is location. These are admixture components. So when you when you have reference populations, you can calculate admixture components of the typical Chinese, typical Russian, and typical Tatar. And of course, it's a gross simplification because all Chinese, there are so many different ethnic groups within China, so many different ethnic groups within Russia, and also there are different groups of Tatars. And yeah, so when we create typical population, we are making, again, gross simplification. But it's, yeah, sure, Amanda?
I'm just not familiar with the previous, I think maybe 2 ago. What do the colors represent on the?
Okay. So in this case, the colors are very. So what we can do is, it's just for visualization. We can since we represent the person in this say like a 9-dimensional space, we can also assign a color. So Northeast Asian, Mediterranean, they're all different colors. They're just for ease of representation. Okay. So visually, it's easier to look at composition of colors than just look at composition of numbers.
Okay, so like if two share the same color, then that's like a similarity.
Yes, yeah. So because if you look here, right? So we can also look at numbers, but it's easier for us to look at colors. Okay. Because one of the first rules of bioinformatics, you need to look at everything using your eyes, just just examine the way the data look. And here it will be easy because it's much easier than just look at the columns of numbers. Looking at colors are easier. So for example, here, if you change here from this like a dirty green to red and blue combination, you can assume that we moved from one group of ancestries to another. Okay. So these people are not supposed to be the same, and so if they are labeled the same, there's some mistake. Okay.
This
And
hurts
just
my eyes.
sorry.
It's so bright.
Okay, so and if you test this approach using some reference populations, what you can see that in some populations we can reconstruct their label very accurately. In some cases we fail. And for example, for Finnish people, success rate of reconstruction is very high. It's actually above 80%. It's close to 100%. And in some cases, from Kuwait, we are not very successful. So we can also, if you take for example, do leave one out validation, we remove one Kuwait individual, we cannot reconstruct the person is Kuwaiti. Why do you think it's happening? So Finns are very easy to reconstruct, and Kuwait are very hard.
Well, I guess depending on mating patterns,
And
that could be a factor.
also,
Access to the individuals or to that data?
maybe, but it's not the major component. So what do you know about a lifestyle in Kuwait and a lifestyle in Finland?
Oh, very different, I'm assuming.
Yeah.
Yeah, it is very different. Yeah, so Finnish population, until recently was maybe quite, docile, and they lived in a certain community. They didn't move around much, right? So
Versus like a nomadic,
yeah,
constantly, okay.
yes, and because it's Middle East, it's hodgepodge, and in addition, all the boundaries there, they were done basically using a pen and pencil, right? Okay, this is your kingdom, that's my kingdom. So and it's still very politically unstable. So for example, imagine some Bedouins, and they're on both sides of the border, the same people, right? They're just they totally ignore the borders.
Right.
So, and there are not many people are nomadic in Finland just because it's very cold. So it's very hard to be a nomad.
Yeah.
So, and it's again, the lifestyle actually, is responsible for accuracy of the prediction. So for places where people stay in the same place, they don't move around much, they're very easy to identify using those methods, just because they really become very attached to the place, very typical. But if people move around, and for example, around Mediterranean, people were moving around Mediterranean, and that's why people from Italy would be more similar to people in Greece. And even the distance is large because of the common play, because they were moving around using boats for thousands of years, and of course they were exchanging DNA when doing it. So we have to take into account population history and in the lifestyle. So but still, so it works. But what do we need to do? So people report to us, so they are ancestries. So let's say we have collected some samples. And say if you have 10 individuals that were reportedly from the same ethnic group, okay? So my suggestion is don't trust the labels, okay? So why I suggest not to trust the labels? Because frequently people have no idea who they are. So first of all, infidelity. So people tend to hide that they were unfaithful. So that's, so second, there were adoptions. In some societies, adoptions are kept a secret. So children are not told they're being adopted, and they can be adopted from totally different ethnic groups, but they were raised as local, and in addition, there could be more traumatic events, and and that wars, and all kind of unpleasant things that leave the imprint in DNA. And so here, for example, we are looking at some Native American population and collecting 10 samples. What we see that we can cluster the vectors, because of course we cannot cluster the huge genomes, but as soon as we got also to the small, well-defined vectors, we can cluster them and we can visualize them in a nice bar chart. So we see that 5 of them are very nicely clustered around Native American group. Then have another cluster that's marked in red. They have a quarter Mediterranean, and the rest is Native American. And we have one more cluster of 2 individuals that has slightly more Native American and less Mediterranean. So so in this case, my gut feeling would be not to use red and yellow group because they're not typical. So for us, if you want to do population studies, typical people are those. Not these guys.
So if you were to recommend to not look at the labels, I guess in order to make sure that they are telling the truth, why don't scientists take that extra step in making sure that whatever you're describing yourself as is actually true?
So that's the only way to do it, because in some cases people look the same. They are not supposed to, to look different if they're being adopted. They may be adopted from a different tribe, different ethnic group, but they phenotypically may be the same.
Right.
So, and also when you come to study new ethnic group, you don't know how they're supposed to look. Maybe there is a intrinsic variability of phenotypes. So this way is the only way to see what is happening. And frequently people don't know. So these people honestly report on themselves as true native people that have, and they do have the culture, they do have the language.
Right.
And for all parameters, they are this group. They just don't have the DNA. Okay. So, and of course, that's why if we take all of them into account, so we already spent thousands of dollars sequencing them, right? So, and now what we have to do, they have to lose these guys. Okay. So for our analysis, we only have 5 individuals, and it may not be enough. Okay. So we and this is pretty easy, because you only lose half. Sometimes you have, okay, I only have 1 individual left, but you cannot do statistics of 1. Okay?
Right.
So if you approach Ancestry globally and you have to discard admixed individuals, you are losing a lot. But look here. So these guys, they do have 45 up to 82% of the genome from the group that you're interested in. Okay, so if you use a local ancestry approach, you can still use their DNA. So, but you can just can't do entire genome. You have to cut parts of the genome that you can use. Okay, otherwise you're losing half, and we don't want that, because it's a lot of money. And it's a lot of effort. You have to be in the jungle. You have to look for this tribe. You have to negotiate with the chieftains. Please let us take the DNA, and it's dangerous, and it is it's challenging, and it's physically very demanding and financially very demanding. So you don't want to lose half of the samples. Okay. So and doesn't matter. It doesn't have to be humans, right? It can be any population of, it can be animals, it can be plants. And so in this case, you skip the negotiations, but still you may be wasting money in carrying the samples, and it's still quite physically demanding. All right, so then we need to work on some more elaborate methods, and how to look at those people. So another method that we have developed is called readmix. And in terms of computer science, it's very involved method. But what it boils down to something very simple, is how we want to represent the person as a linear combination of say several ethnic groups. Say you want to represent the person as a mixture of, say, British, Russian, and Chinese. Okay, but whatever the components are. Okay, so why they want to do it? Because it's going to be your first step. Should you even bother about local admixture? Is there a linear combination? Do you have any data in your database that you can partition the individual? So of course you assume there is some error, and so doing this, we tested it on several interesting samples. And for example, we looked at Bronze Age Europeans. And so of course, as you know, that if you look at Europeans, you can they came as a combination of hunter-gatherers and Neolithic farmers. And hunter-gatherers mostly lived at the north of Europe, and farmers mostly south of Europe. And if you look at the samples, so for example, samples from Tuscany, from they are more farmers, like 60% farmers and 31% hunter-gatherer. But if you look at Britain, you get the reverse relationship. There are 60% hunter-gatherer and 40% farmers. And of course, these two groups were, of course, mating and exchanging DNA. But even if at the Bronze Age, or maybe 5,000-6,000 years ago, you see the signals of different groups populating Europe. And of course, you can see it also in the signals of modern Europeans. So, and this is the way the map looks today. So the darker color shows the hunter-gatherer ancestry. So that's mostly limited to southern part. And because, as you know, the farming. So girls, do you remember where farming started? So what
you said where does it start?
Yes, so with the cradle of farming.
I cannot hear. I'm missing like one word.
Okay, so where was the farming started? Was the cradle of farming?
I don't know.
Forgot.
Yeah.
Dallas.
It was like, somewhere here in Anatolia.
Oh,
Anatolian
yeah.
farmers. Remember Anatolian farmers? Yeah, that was there. Okay, so these are the guys. Okay, and they gave their DNA to Mediterranean, and it's also spread and all the practices. Okay, so and of course we can apply it to modern individuals, and so as you can see, if admixture profile is showing differences, we can also see different admixtures in using readmix. If in a group the admixture profile shows everyone as identical, you see we make one prediction for everyone, and so if there's a slight difference, we also represent as a different mix. Okay, but now we want to go to make maps like this. So for our clusters, the one I showed you before, we want to represent our individuals using those maps. So look here. So remember cluster one, there are five individuals that look like the perfect Native Americans. So and that's the way they look. They have a Native American DNA everywhere, right? So cluster 2 and cluster 3 were mixed clusters, and cluster 2 more so. So you can see piece of Mediterranean, Northern European, and other influences in different portions of the genome. Okay, so that's but what can you do for subsequent analysis? For example, if for studying of loci, you take those. Then from these guys, if you look at the first chromosome, you mask out the different colors, but you can use it, right? So here, chromosome 17, use it entirely, right?
Yeah.
So you just remove the colors that you don't like, and you do your studies.
So chromosome 20 and 21, you'd also be able to use in cluster 3.
So 20. So in this case, you will be able to use them, but you will be able to use only parts of them.
Okay.
So you can use your scissors, right?
Mm-hmm.
So if you're interested in the locus that is located here, then tough luck, you will not be able to use this cluster at all.
Right.
But if you are interested in something that's located here, then you're good. You can use cluster 3, but you will not be able to use cluster 12 because here you see the influence for other people.
Mm-hmm.
Okay. But in this case, you have to be careful, and depending on what genomic vicinity you study, you either use all of them, or you use only those that have the ancestry that you need. Okay. So, and of course, where statistics comes. Okay. So the approach that we used, it was a Bayesian approach. Okay. Okay. So so first of all, so do you remember who was the Bayes? The old Bayes guy. So
Bayes theorem.
Yes, so Bayes theorem. Yes, that's what we see. But what do you know about Bayes? Who was he?
Wasn't he the famous mathematician? Remember, we were talking about him in stats. He was the famous mathematician. Blah, blah. No. Oh my gosh. I don't remember.
He was a smart guy, but he was not a mathematician.
Oh, my bad.
What was his occupation?
Is he a biologist?
No.
Statistician?
No.
Wouldn't
Was
that
not
be a
a
mathematician?
No, he was not a mathematician.
He had something to do with stats. I know that.
No, he has friends who were statisticians, but he was not.
Shoot.
Is he a physicist?
No.
No. Was he a musician?
I'm shooting blanks. Sorry.
He was a priest.
Oh,
I don't remember this one.
So the guy was a priest, and he was, but he was very philosophically minded. He had many friends who were mathematicians, and actually he was just musing about, assuming you want to go to heaven, what's probability to have angels at the end of the needle, all kind of musings, and probability of entering in eternal life, giving you have all the wealth. I don't know, all kind of philosophical issues, but he was formulating it using this conditional probability principles, and then I think his friends formalized it, and but it wasn't based. I think so. As far as I know, it can be historical anecdote, but he hasn't wrote a single formula in his life. That's it. I think.
I do not remember that story.
Okay, hopefully you remember it now. All right, so of course, so what do we know? So even we have people of, we have our zombie populations, right? So we have our zombie populations, people that based on people of validated ancestry, right? So given that we know that somebody's from certain population, we can calculate the frequency of a genotype, okay? But Bayes theorem is doing things the other way. What's the probability of belonging to a certain population, assuming you observe a certain genotype? So basically, because Bayes theorem is just inverting things, right?
Mm-hmm.
So probability of a genotype given population, and what you want probability of population given genotype, right? So you're just switching things around using the Bayes theorem, and that's what it is. So you have multiple populations, like source populations, zombie populations. You call them ancestral populations, and you know what kind of probability of a genotype, and you can calculate it. So we have this probabilities, and then what you do, you calculate this matrix, and of course, you do it in a log scale. For computation, because it's easier, and then what you want to do, you want to if you do computer science or any kind of algorithm, you try to solve optimal path problem, because what you want to do, you want to find an optimal sequence of states along the genome. Okay. So you go from, say, Mediterranean, Northern European, African. So, and you want to find, you're trying to find a path through this matrix that gives you the maximum score. You optimize.
Oh, so that was the one we did up and down, left and right diagonal, right?
Yes.
Okay.
Yes. That's the first we did this. Yeah, that's good. Good. Yeah. So and if you want to formalize it, you have this recurrence equations, and so first of all you build the matrix, and then you do remember the trace back, right?
Mm-hmm.
And this is what we do. You do it for every chromosome, and you recreate the sequence of states for each individual, and or for groups of individuals. So as a result, and of course, using colors to represent different ancestries is easy, because it's not numbers. Numbers are hard to see, but if you put colors on a plot, you immediately see what is happening, and that's a result of our traceback approach. When you calculate local ancestries based on those probabilities, and so, but the next question is, how would you use it? Okay, and in this case, we have to go and we have to refresh what databases are containing information about the function of genes. So, are you familiar with the KEGG database?
I don't think so.
Gene Anthology, GO database. Possibly.
I think we might have used it.
Yeah. So there are several databases that group genes together based on the function that these genes are involved, okay? And not just function, you know, processes, cellular components. And so what you can do is you can calculate, because you have your whole genome sequence, right? And for example, now you have marked all the individuals with certain ancestries. So for example, you have Asian, Northeast Asian, and you have Southern European. And you want to compare them and see what kind of adaptations these guys are having. And people mixed a lot, especially Mediterranean people are mixed, and Northern European people are all mixed. But now you did your job. You did the previous step. You have divided the genomes into windows, and now you have removed from all your collected individuals. You removed all the areas of the genome that can be attributed to later migration, or to whatever process that you don't want to attribute to adaptation that happened in this area for thousands of years. Okay, so you're just removing the last 2 centuries of infidelity, right? So you remove them. Okay, so you only kept those people that they are adapted to these areas for many years. Okay, and then you calculate, or when you contrast 2 populations, you calculate for each pathway, which functional group, as you say, how many also a total number of non-synonymous changes. And why we are looking at non-synonymous changes? Why not just all changes? Why we're interested in non-synonymous changes? Can you speak on it?
Let me think it through. 1 second.
Why not all SNPs, but we are looking specifically at non-synonymous SNPs?
Because if they're changing at the same time in the same location, then it won't be like indicative of different regions, right?
No, it's the same location. So we're looking at same locations, but so what you see like all kinds of, because what is non-synonymous SNP? So so what's a synonymous and non-synonymous? So
it's not, wouldn't it be in the same part of the chromosome?
No. It's they're all part of, say, maybe on the same gene, right? For example, right? It can be, yeah. But some changes are synonymous, some changes are not.
Non-synonymous is when the amino acids aren't altered.
So non-synonymous when amino acids are altered. So if it changes non-synonymous, you change the amino acid, right?
Right.
Yeah.
Okay.
So, and when you change the amino acid, it means your protein is different.
Yeah.
Okay? And means already because the function is functioning differently. Okay. So, and if you have lots of changes in, so for example, in Eskimo compared to Italians, in a say, and in a pathway that's related to temperature regulation, you can sort of speculate it's because Eskimos live in a much colder environment than, say, people from Italy. Okay. So, and that's why you group by pathway, by function, and you're trying to see if it makes biological sense. Okay.
Okay.
But if changes are non-synonymous, they don't change the protein. They don't manifest phenotypically. So because we are looking for the changes that can be linked to a function, that can be linked to phenotypic manifestation. Okay.
Yes.
So so that's why what we try to do, we try to calculate the relative between, because we compare 2 populations, and we're trying to compare the frequencies of non-synonymous changes. So for example, if you have population A and population B, right? And we have overall for, we take all the pathways, everything, right? So we know how many total non-synonymous changes in population A, total for B. And when we try to analyze different pathways separately, for example, this temperature regulation, so we calculate how many changes in A and total A versus B. And then we can calculate the score. So, and in this score, we are changing, we are assessing the for particular pathway, so the frequency of non-synonymous changes versus the background level. And the difference between the frequency in a particular pathway and the frequency, that expected frequency, is going to tell us, look, in a sense, it's just a z-score. How significant is accumulation of changes is in this pathway. So, and of course, what we need to do, we need to scale it. We need to compare it to the synonymous changes, because if two populations accumulate a lot of synonymous changes, then accumulation of non-synonymous may not be as significant. So we want to only look at changes in non-synonymous positions, and but if synonymous changes are not there. And in this case, we can compare all pairs of populations. So, for example, this is Siberia and this is Eastern Europe, and everything that's if the same in the same pathway, we have synonymous changes are drastic, we discard the pathway. But if in the same pathway synonymous changes are at bay, at zero level, but non-synonymous changes are huge, these are interesting cases. So, for example, we can look at what's important in say in Northern European. In Northern European, one of the most interesting cases that come up is the olfactory pathway and in the olfactory receptors. So, this result appeared multiple times. It appeared when we compare ancient people from Bronze Age to modern Europeans. And if you compare people who live, say, in Siberia, people who live in Latin America, all the native tribes, we compare to Northern Europeans. So what's happening that Northern Europeans are losing the sense of smell. So, and this is an absolutely crazy result. And if you look at the birth of Europeans, most Europeans accumulate the largest number of mutations with a huge significance in the pathway related to olfactory transduction. Can you speculate why Europeans are losing their sense of smell? Especially if you compare, say, Bronze Age versus modern.
Right.
Like we're breathing in chemicals that makes us lose our smell.
Mm-hmm.
I think the way it works in selection is if you breathe in chemicals, yeah, but so we don't need it. We don't rely on it. So people who live with the nature, right? They need to rely on the noses for many things, because we need to, if you live, say, in the forest, you have to smell the leopard coming to you, right? So you rely on it for many things. But if you live in cities, and if you're in civilization, then it's you don't need the acute sense of smell. You're fine without it.
Right.
So that's one of the artifacts of civilization, where you lose your olfactory transduction gene. So of course, everything looks great. We have amazing methods, but let's decide how can we test if actually the method makes sense. So let's think, how would you, girls, how would you decide if the method actually works?
You test it. Well, first off.
There is no gold standard.
All right.
Let's come up
with which part of it.
Yeah, so how can we test? Of course, our prediction that, this area is Native American, this area is Mediterranean, this area is North American, this area is Near East. How would you do it?
You would probably do it based on, so if you're looking at certain traits, so going back to how Northern Europeans are starting to lose their sense of smell, then you would simulate some type of experiment that tests that theory.
Yeah, so one of the, yeah, you can do sort of phenotypical, yeah. So if, for first of all, if results are not making, if they're not looking silly, like at the previous slide, yeah. So you obtain results that, supported by medical literature, something that you already know.
Right.
Yeah, another result, but purely computational. So if you remember, we started with a very crude description with admixture vectors, right? So, and as you know that they give, person say in component, it can be 9 component, 13 component, 25 component, that they represent as a vector, okay? So what would be very desirable if you add all the components from the vector that, all the local ancestors, right?
Mm-hmm.
If they add up to the same global number.
Yeah.
That would be very nice, okay? So if you, if you look at separate pieces taken together, you get the same global picture. It will show some sort of stability. But of course, they may be, add up correctly. But if the sum is equal to the right number, it doesn't mean every term is correct, right? But still, it's encouraging at at the absence of other evidences that may be good. So what we did, we computed using 1,000 genomes, and it's supposed to be gold standard data, and we compute the correlation coefficient between what is we calculate as a global ancestry using admixture, and when we add up components along the genome, and we get very nice distribution. It's almost 1, right? And if you at the Euclidean distance between the vector, that admixture vector, and what you get from putting together components from local ancestry, you do get the distance that's pretty tiny, okay? So you are very close. So in this case, I'm cautiously optimistic that the results are there.
So, oh, sorry, can you go back?
Go ahead. Mm-hmm.
So the ones that are having like the higher distances, like over here where it's 0.05, 0.075, you're not as concerned about those?
I would, yeah. So they're not huge, yeah, but so I am much more happy about those. And of course, there are always difficult populations. There are always difficult samples, and it's important to know who they are. And so when we make a prediction, and we know that, oops, yeah, kind of in our unfortunate areas. So our confidence in the prediction for people who traditionally fall here would not be very good. But it's still small. But you know that if, for example, if somebody is here, right, for those people, then yeah, you would be much more confident. But as you remember the original picture, for example, Kuwait, right, people. If I think it's some areas are difficult, and we know that some experiments are harder than others. Okay. But it's important to be aware of it. It's important to know where you can succeed and where you are not expected to attain perfect results. So so we did this computation for multiple subsets. And so what are our conclusions? So modern people are admixed, okay? Some people are not aware of it. And people can tell you I'm pure X, but the person can be pure Y, or admixture of XY, or admixture XYZ. And they're not lying to you, they just don't know.
Right.
And if you are making personal medicine, for example, it's very important to know, for example, if you have like susceptibility locus at some position, right? So you may think you are A, but in this locus, you are B. And this medicine cannot be given to you, okay? So of course, it's still visual thinking, it's still looking forward to in the future, but it's very important to know who you are at different positions of the genome. And of course, it's just very, very interesting. Yeah, it's not just medically and important in terms of population genetics. And so if you look at use a local ancestry method, not necessarily this one, but other methods, we can classify genomic regions based on the ancestral origin. And what helps save money for researchers. Researchers can now collect samples from cities. And because we know people in cities are mixed, and then we can remove everything got admixed in the last few generations, and keep portions of the genome and increase the power of our analysis. So that's kind of the research. And do you guys have any questions?
Not a question, more of a comment.
Okay.
Just like it makes, especially the last point, it makes sense how useful this is, because even in my experience, like when my mom did her genome analysis, like she had different things that we never even considered would be part of our DNA or part of her DNA based on looks alone. So
Yeah.
like in the United States, where it's just a huge melting pot, or especially in like suburban and more modern areas, like I understand how people will look at each other and think, oh, that person is just from this one region ancestral, in their ancestry.
Yeah, and we are so much more than, you know, that's you know, it's a label, right? We are of labels, and I think that's you know, understanding that you know, people are so complex inside and out. I think that's you know, it's very, very important.
Yeah.
Okay, girls, do you have any other comments, questions, suggestions? No. Okay, so then I'm going to stop the recording.